Welcome back once again. This chapter, chapter 6, will be about metabolism. And that's a word we've probably all heard, but we may have different concepts in our minds, what it means that might be different from what it means in the context of a cell biology course. Metabolism in this chapter, and in terms of cell biology, refers to every single chemical reaction that's happening in a cell or in an organism. So it's not just about how quickly or how slowly one processes food, the rate of your metabolism. We're talking about the sum total of all of the reactions. The hummingbird gets put here as an example of a creature that does have a very high metabolism because their wings move so quickly to allow them to hover in flight. They have very high energy demands. So they have to be very, very efficient with their metabolism. This chapter is going to be talking about some of the types of reactions we will see in metabolism. And in the next chapter, we will get into the specifics of how we take certain high energy molecules, break those bonds, and extract the energy from it. This chapter is going to talk about forms of energy and the enzymes necessary to do those types of reactions. This chapter is also the end material for our first exam. That will cover chapters 1 through 6, and it will be on Wednesday, September 23rd. The study guide will be getting put up by Wednesday, the 16th. A very cartoon and abbreviated way of showing energy flow. On our planet, all of our energy comes from the sun. In our solar system, all of our energy originates with the sun. And that energy can be transformed in several different ways. Photosynthesis can harvest light energy and convert that into chemical energy. Those are the photosynthesizers. Plants are listed here, but plants are not the only ones that photosynthesize. There are many different types of algae that photosynthesize, and there are even bacteria, some prokaryotes that can photosynthesize. And herbivores and omnivores eat plants to obtain energy. Carnivores and omnivores eat the herbivores, and the decomposers digest all that matter. None of these energy transformations are 100% efficient. There's always some energy that will be converted to heat energy rather than to chemical energy. And so that energy isn't lost, it's just in a format that is not directly chemical. So the producers, the photosynthesizers, will harvest sunlight energy and turn that into chemical energy. Consumers, not just animals, but also bacteria and worms and other creatures. And the decomposers will digest the dead producers and consumers, and that is eventually also going to convert all that chemical energy to heat. If any of you ever do backyard composting, whether in a compost heap or in a tumbler, a lot of heat gets generated in that decomposition in a compost heap. I lived in Minneapolis for 6 years during graduate school, and we had a compost heap in the backyard of one of the places I lived. And in the dead of winter, when it is below zero, there was no snow on that compost heap, and there was steam rising out of it. So we'll keep that in mind when we get into our specific metabolic pathways in Chapter 7. We're going to show balanced chemical reactions and keep track of all those atoms that are going in and coming out. And we will show energy, but we don't ever show the heat. And so that's always something we need to keep in mind. So we have plants like our tree doing photosynthesis, so our little fat squirrel has something to eat. And that is true for any circle of the carbon cycle. The producers have to harvest the sunlight, the consumers are able to take advantage of that, and the consumers create wastes like carbon dioxide and water that the producers need to be able to create those carbohydrates. A very, very rough phylogenetic tree where the prokaryotic ancestor, that of course we don't know because no one took really good lab notes back then, but that original ancestor had to have anaerobic metabolism because there wasn't sufficient oxygen in the Earth's atmosphere. So anaerobic means without oxygen. There are still organisms in the archaebacteria and the eubacteria that can definitely live in the absence of oxygen. Some of them are able to do so if they have to, others are required to do so. Once we get above that, all of these organisms have some requirement for oxygen. So metabolic means all the chemical reactions. We're going to be looking and breaking down our chemical reactions always into sort of a dichotomy. It'll be this kind of reaction or it'll be that kind of reaction. And so if we look at the biggest, broadest categories of metabolism, we can have anabolic pathways where we take small things, provide them with energy, and build large molecules. So energy is required. Or we have catabolic reactions. We start out with something bigger and we break them down into their smaller components, and that releases energy. So anabolism or anabolic pathways are often generated through dehydration synthesis, where we can remove a hydroxyl functional group from one subunit and a hydrogen off another, remove the water, and build that covalent bond. And if we're going to build a covalent bond, we have to put energy into that system. So we input energy, and that decreases entropy. Think way back to your Chem 1A class. So entropy is a measurement of randomness in a system. So we start out with something that's fairly random and disorganized. We provide energy, we create something more organized. We decrease the entropy. With catabolic reactions, we start out with something very organized. We carry out those hydrolysis reactions and break it down. We release energy and we increase the entropy. We make it more random. I remember the difference, which one's anabolic, which one's catabolic. I learned it because catabolic has the same prefix as catastrophe. Things are falling apart. And so anabolic's the other one. But you may also have heard the term anabolic steroids. So hormones that can be used to bulk up, to build bulk. So anabolism, you get bulky. Catabolism, things fall apart. When we talk about types of energy, we already talked about light energy, sunlight energy. We talk about chemical energy, but we also have potential energy and kinetic energy. So potential energy is stored energy. Kinetic energy is the energy of movement or work. Not as relevant in cells. We don't have dams built up in our cells, but we do have chemical potential energy. Whenever we have covalent bonds, that is stored energy where those shared pairs of electrons are, so that if we break that bond, we can release energy, and that energy can be used to move molecules around and be kinetic energy. So here's our chemical energy. This is gasoline. We can convert that chemical energy into mechanical energy, and that mechanical energy into kinetic energy of moving a vehicle forward. You've probably seen on a gas pump the term the octane rating, and that has to do with how many of these hydrocarbons containing 8 carbons are present. So high octane gas has a greater proportion of the octane gas form as opposed to different length chains in the hydrocarbons. When we look at our anabolism versus catabolism, anabolism energy in, catabolism energy out, we also call those reactions endergonic because they require energy in, and exergonic, energy out. So anabolic reactions require energy. They are also endergonic, energy in. Catabolic reactions release energy. They are exergonic. The other aspect to whether a reaction is exergonic or endergonic has to do with the Gibbs free energy, which is also looking at the activation energy. What is the energy difference between the reactants and products? That will be our Gibbs free energy, and how much interaction has to happen in order for a reaction to start at all. That is our activation energy. Here we're looking at the Gibbs free energy. The difference in energy between the reactants and the products. On the left says it is an exergonic reaction. The reaction is spontaneous, and that our change in the Gibbs free energy is less than 0. It is a negative number. This triangle is the Greek symbol delta, and it is used to represent a change in. So delta G is the change in the Gibbs free energy. Whatever these reactants are, once they complete their reaction, the products have less free energy. So we have released energy during that process. If we release energy, it is catabolic, and it is also exergonic. The delta G is negative. We end up with less free energy in our products. We have the same amount of energy change in the image on the right. However, this reaction, the products have more energy than the reactant, meaning we had to put energy into the system. If energy is added, then that's not spontaneous. We can't just have these reactants in place and expect the products to form without energy input. And so the change in free energy is greater than 0. It is a positive number. So if you have a negative delta G, as in this case, where the products have less free energy than the reactants, we have released energy to the environment, it means the reaction is energetically favorable to go toward those products, but we might need a catalyst. We might need some sort of surface, something to allow those reactants to be in the correct position in order for that reaction to happen. Here's that activation energy I mentioned on the previous slide. This is still an exergonic reaction. We have a negative free energy. It is spontaneous, or is capable of being spontaneous, but we have to overcome this activation energy in order to get to those products. And so an enzyme or a catalyst is required. And without a catalyst, we have very high free energy or activation energy to overcome. And so if we don't provide the catalyst, even though this is energetically favorable, these reactants are not going to interact and will not create a product. If we provide a catalyst, then they are much more likely to overcome that activation energy and be able to proceed toward products. In an inorganic chemistry class, a catalyst could be a number of things. It could be heat. It could be some sort of metal. There's lots of different things that can help a reaction to proceed. In a biological system, the catalyst is almost always a protein, and it is called an enzyme. Heat is not a great catalyst in a biological system. Biological systems tend to have a very narrow temperature range under which they will perform well. If it gets too cold, as in even a test tube, the molecules don't have enough energy to react. If it gets too hot, most of the chemicals that we require to get our reactions to move are proteins. And if proteins are overheated, that secondary structure and that tertiary structure tends to sort of melt because the proteins are held together by hydrogen bonds, and heat can easily disrupt hydrogen bonds. So transformations of energy, we already talked about light energy to chemical energy. We saw the chemical energy of gasoline being converted to mechanical and kinetic energy. But we can convert in different means as well. We can have chemical energy can be converted to light energy. Now, plants don't reverse photosynthesize, but there are organisms that can do bioluminescence, like lightning bugs or fireflies. They glow in the dark. There are jellyfish that produce fluorescent proteins. There are microbes in the water that can also do that. So energy can be converted between different forms. It cannot be created or destroyed, but we can convert it in various ways, and it's never 100% efficient. We're always going to have some of that energy being converted to heat. There's our entropy, the measure of randomness. So gases have higher entropy than liquids. Liquids have higher entropy than solids. So the greater disorganization you have, the higher the entropy. The more organization you have, the lower the entropy. This giant molecule, which you will never be responsible for drawing, but should be able to recognize, is adenosine triphosphate. There's 3 phosphate functional groups. There is a ribose sugar that is the exact same sugar that we see in RNA. And then this nitrogenous ringed structure is the adenine. So this adenosine triphosphate is also used in building RNA. It is the energy molecule that allows, if we lop off 2 of these, this is a nucleotide. The ribose, the nitrogenous base, and the phosphate. But it's the hydrolysis of these phosphate anhydride bonds that releases energy for the cell to do work. So ATP is our energy currency, our primary form of chemical potential energy that we can use to do work in our cells. We saw the sodium-potassium pump in the chapter on our membranes, and we saw the ATP there. Energy coupling, because we take the energy that we got from the ATP hydrolysis to pump the sodium-potassium ions across the membrane. Because we maintain and continually pump sodiums to the exterior of the cell and maintain high sodium outside, and we take potassiums from outside and pump them inside to maintain a high level of potassium inside. And so we keep needing that cellular potential chemical energy to drive this pump and maintain that difference across the membrane. Those enzymes that I mentioned that can lower that activation energy provide a surface. They are not a reactant. They are not a product. They are the surface that allows the reactants to come into the correct position so they can interact to create the products. And so they can speed up a reaction rate, but they cannot change the free energy change. There will always be the same amount of difference of free energy between the reactants and the products. We can't change that, but we can change how fast we get there. So the enzymes get reused over and over again. This is a representation of how enzyme recognizes the chemical it interacts with and what that process is. So the enzyme is the gray blob. The substrate is the reactant, the chemical coming into the system. When the substrate binds to the active site, there is a conformational change, a temporary physical change in the enzyme to fit more tightly to the substrate. That is called the induced fit, because that tight fit is induced or caused by the binding of the substrate. And that allows the enzyme to apply mechanical energy pressure on the reactant, on the substrate, and create the products. The enzyme is then returned to its identical shape it had at the beginning and can get used over and over. So the enzyme is not a reactant, it is not a product, it is not permanently changed during this process. There might be temporary changes in it, but there's no permanent change. And depending on the reaction and the enzyme can be carrying out this process from dozens to thousands of times per second. We'll be doing an online virtual enzyme activity lab where we will look at the rate of reaction. This is showing in general, as the substrate concentration increases, the rate of reaction also increases. And with a normal enzyme, it will increase to a certain point, but if we get a high enough substrate concentration, we saturate the system. There's no way we can go any faster if we add more substrate unless we also add more enzyme. We are never going to get a faster rate. It will flatten out. If we have a competitive inhibitor, that is a molecule that, at least on part of it, has a similar shape to the substrate and competes for that active site. So if you have a competitive inhibitor present, but a lot of substrate, you can have the substrate outcompete the inhibitor and still get to the maximum rate of reaction. If you have a non-competitive inhibitor, it is binding outside the active site. That distorts the active site and the substrate can't bind. So we have distorted, mucked up enzyme. So even as we add additional substrate, we can't really overcome the effect of the inhibitor messing up the active site. When we look at that non-competitive inhibition, it is also called allosteric inhibition. Allosteric means a different site, a different placement. So the enzyme has its active site, and it has another position on it called the allosteric site. The inhibitor doesn't look anything like the substrate. It's not competing for that active site. It has a different site where it binds, and when it does so, now the active site cannot bind the substrate. We could also have allosteric activation, where the initial enzyme has the altered active site, and once the activator binds, then the active site can fit the substrate. We need the competitive and allosteric inhibition because we do not need every single reaction in our cells being carried out all the time. We have to have natural ways of inhibiting or slowing down those reactions so the cell can function normally. Likewise, there are some enzymes that are rarely needed, so they will remain in this inactive state until something comes along to signal the cell that it does need to carry out that reaction. The question, have you ever wondered how pharmaceutical drugs are developed? You very well may not have wondered, but it has to do most often with enzyme pathways. If we have some sort of microbial infection, a good drug target will look for some sort of metabolic capacity that the microbe has that is not found in us. So if we can specifically inhibit a microbial enzyme that is necessary for their life, then we can disrupt that microbe's capacity to metabolize and do no harm, or at least less harm, to ourselves. There are also coenzymes. Vitamins are either coenzymes or precursors to them. They are needed to increase that enzyme activity or efficiency, and there are several examples here. You do not need to memorize the structures or even which vitamins are coenzymes, just that vitamins are coenzymes. This is showing how that allosteric inhibition works to inhibit pathways. This triangular substrate, whatever it might represent, fits perfectly into the little Pac-Man enzyme, converts that to the first intermediate. That intermediate is now a substrate for the second enzyme. It gets converted to the next intermediate, which is the substrate for the third enzyme. That creates a product. That end product feeds back to the original enzyme and now alters that active site. So this happens in our metabolic pathways when whatever this is, if it's in abundance and is useful to the cell, can be converted to this until the cell now has a sufficient amount of this end product. When that is insufficient amount, it will go back and tell the original enzyme, we don't need any more of this. So that is one way of an allosteric inhibition being regulating an entire pathway. So we don't have to shut down every enzyme. If we just inhibit the first enzyme, we never create this, and the rest of the pathway does not proceed. All of that will be necessary for getting into Chapter 7, where we talk about specific metabolic pathways. We will see that activation and that inhibition. We will see the different points on pathways where that enzyme regulation will be important. But this chapter, Chapter 6, is the end of the material for your first exam. I will have a detailed study guide that will be posted to telling you which concepts to focus on, provide you a vocabulary list, provide you with some thought questions, and all of the details in terms of how you will go about taking your exam through Canvas.